So we've seen those molecules of chlorophyll being important for the process, talked a little bit about how the molecule functions on its own. And here we have now a little more zoom out into the parts of the photosynthesis, photosystems as they call them, these complexes of pigments and proteins that make up are found really densely through the membranes of the chloroplast. So down here we've got an illustration of a chloroplast, just reminding you that you've got these stacks of membranes. They're sort of like membrane pancakes. And membranes are important to this process. They have an inside and an outside. And the inside has this name of lumen, so sort of the inside of this pancake. And stroma is a fancy name for the juice inside of the chloroplast, but outside of those stacks. So lots of terminology. I'm trying not to emphasize too much the terminology of these different structures. I'm trying to get a little more at the heart of what's a little more important. So photosystem, as I said, proteins. So proteins are made through genes being decoded into proteins, and then also a whole bunch of pigments. So these green disc looking things through here, these are stand-ins for pigment molecules, chlorophyll, and related pigments that are responsible for gathering light. So photosystem is a really good name for these. They're responsible for collecting light, and then the protein puts everybody into the right arrangement where that light energy gets directed toward the right place. Shown at the left here, we've got one called photosystem 2. And photosystem 2 has this so-called reaction center where like the most important chlorophyll A molecules are found. But then a whole bunch of other surface area for collecting light energy and passing it on. The two photosystems, they are distinct and sort of complementary. They help each other out. And photosystem 2 is the one with this reaction center. Photosystem 1, we'll see that function in a related fashion. And as I said, these would be found very densely throughout the membranes of the chloroplast thylakoids. All right, so here's a throwback to what you probably learned in introductory biology. I think it's, maybe if you haven't had it for a while, it's good to see this again. It's a really cool process. I think it gets kind of bogged down by this need for students to memorize all the parts of it. I don't know how it's being taught these days, but again, I'll try to give you what I think is a bigger picture overview of this and show you what's going on. So the PS2, PS1, those are the photosystems from the last slide that I showed you. And we see a little bit of what we've seen before where the water molecule gets split and releases an electron. So in the presence of light, that allows a water molecule to get split and release an electron. And the electron has some energy, or is sort of conveying some energy to the system. And it bounces along these other molecules, so cytochrome and other molecules, making use of this electron molecule, passing it down the chain. Photosystem 1 also responds to light. We saw that it was abundant with pigment molecules. This is not responsible for splitting the water molecule though. So this was not one that had those reaction centers, not doing that important first step of releasing electrons, splitting water, nonetheless passing this light energy on to the electron. So sort of a lot of times this is described kind of like a water wheel. So the light energy is the input, and that energy input allows the electron to move through this process. And ultimately the electron goes up here and allows this molecule to be built, NADPH. So NADPH is sort of an energy storage molecule, and it's going to be used later on in the process, we will see. So big picture, light energy required, water is split, electron bounces around until finally it gets put into this high energy NADPH molecule. All right, so that's called electron transport. These two slides are showing the same background molecules, but emphasizing different things. So this first one emphasizes where the electron goes, and the next one emphasizes the movement of these hydrogen ions, again otherwise known as protons. So as the electron moves through the electron transport chain, it enables this process to happen where protons are pumped across the membrane. And so having this membrane, again, is important. This is very much like a battery. So a battery inside of your little AA battery are two chambers with different charges, and the electrons moving from one charged area to another are what power your devices that the battery is plugged into. So it takes energy to sort of charge this battery. So the electron energy we saw before is enabling the hydrogen ions to get pumped. And shown by the blue dots here, they become more abundant in the inner part. This is the lumen of the chloroplast. It becomes more densely packed with protons, and those have a positive charge. So one side of this membrane becomes more positively charged. And the difference in charge causes the molecules to sort of want to go to where they're less abundant. And they can't just get across the membrane, but they can go through this molecule, ATP synthase. So the protons go through ATP synthase, and through that process, they actually kind of churn it like a turbine and generate these ATP molecules, another high energy molecule. So two ways that high energy molecules are made during this process. In the previous slide, we saw producing NADPH, and in this slide, we are producing ATP. And those will both be useful later. So kind of an illustration of one of these photosystems. So just a whole bunch of protein kind of matrix holding this all together, holding the pigment molecules in the right place. And what's shown with these, I guess the laser beams coming from space, those would be photons of light from the environment. And then these little zigzaggy movements of the light pulses, that's the antenna or accessory pigments passing the light energy between themselves, sort of moving closer and closer to this central reaction center. Pretty cool. Okay, so up to this point, we've been talking about what is called the light reactions of photosynthesis. So the process of getting light from the environment, using that to split water, and then make these high energy molecules of ATP and NADPH. These are all the light reactions. And the light reactions are kind of half of the process, because having those ATP molecules and NADPH molecules, they're sort of short-term energy. They can be used for cellular processes, but they're not great at energy storage. So storing energy as ATP is a little bit not really a feasible option. So sort of taking this energy that's made from photosynthesis and turning that into a stored version of energy, that becomes the next part of photosynthesis, the second part called carbon fixation. So we'll use this term a bit. We've talked about it. We haven't used the term fixation before. To fix carbon is to take it out of the atmosphere where it's really not biologically useful. Take it out of the atmosphere and put it into a molecule where it can be used. So like right now I'm breathing in and out carbon dioxide, and it's not doing anything for my body. It's a waste product. And when I breathe in new carbon dioxide, it's not something my body can do anything useful with. But a plant, again, has this special ability that it can take CO2 in from the atmosphere, and if it has this energy from the light reactions, can suck that energy into a sugar molecule. So down here in the diagram, CO2 comes in from the environment, and using the ATP energy, using the NADPH energy, the CO2 gets put into a sugar molecule. My silly illustration here is of Calvin from Calvin and Hobbes. If you're not familiar with the bicycle episodes, get familiar with them. He had a cursed bicycle with a mind of its own. So this is the Calvin cycle, which is generally how this carbon fixation cycle is known. It's a stretch. Okay, so the process of carbon fixation, again, taking this CO2 molecule out of the atmosphere and taking the carbon atom from that and slapping it into a molecule. This is something that plants can do, but it's hard for just about every other organism to do. Some bacteria have skills in that department. And as I mentioned before, it comes down also to this rubisco enzyme. Rubisco has a longer name, ribulose bisphosphate carboxylase oxygenase. We're going to call it rubisco. So rubisco gets its name by acting on this ribulose bisphosphate molecule. As we'll see, this is already present in the cells, but then they take a CO2 molecule and they slap these together. So we have how many carbons on here? 1, 2, 3, 4, 5 carbons on ribulose bisphosphate, and 1 carbon atom on the CO2 molecule. So 5 plus 1 becomes 3 and 3. So it doesn't go on to make a 6 carbon molecule, but it does make 2 3 carbon molecules. And that is basically the important part, is that that carbon from CO2 is now part of a molecule. And that took energy to do. And that is the building block for bigger molecules. So plants are making these carbon-to-carbon bonds that were not present before. Certainly not present in the carbon dioxide molecule. All right, this one, again, you might have encountered this before and been required to memorize it. I don't ask you to really memorize this, but you might want to be, if you're a person who wants to balance your equations, this is one way to balance your equations. So molecules here differ in the number of carbons that are in their chain. So 3 carbons apiece. This is the kind of molecule that gets exported from the carbon fixation cycle. 3 carbons. Over here, we have an important step where we're going from 3 carbon molecules to 5 carbon molecules. So 5 3 carbon molecules get converted into 3 5 carbon molecules. And then each of those, if we balance this out, each of those can associate with 1 CO2 molecule. And then as we saw before, this 5 plus 1 is 6 carbon atoms. Each of those turns into 2 3 carbon molecules. And it goes around in a cycle. So for every turn of this cycle, we get 3 carbon dioxides in, 1 3 carbon carbohydrate out. So we're actually building molecules that weren't there before, or at least increasing the number of molecules that are there, and uses energy. So we're using ATP, we're using NADPH, we're using ATP again. So the energy that was made in the light reactions can be used to power this carbon fixation process. All right, so again, we saw this before, this diagram of plants, cells, organs, and organelles, and just remind yourself how it all fits together. So we had the light, carbon dioxide, water coming in, sugar molecules produced largely in the leaves, oxygen produced as a waste product, largely in the leaves, and the plants produce sugar. And they also can produce cellular energy. So some of that ATP just goes immediately into plant energy, which is useful for them as well.